Dear students, Assalamu alaikum. Welcome back to Amreena tutorials. Here I am again today, Alhamdulillah, with my 5th video lecture on biomolecules, specifically polysaccharides. Inshallah, we will complete polysaccharides and its subtypes, homopolysaccharides and heteropolysaccharides today. Watch this video till end. Tips and tricks are also included, so this video is going to be beneficial both for your board exams as well as for your competitive examinations. Don't forget to press that subscription button which is popping right now in the bottom right corner of your screen. Press that button so that you will never miss an update from Amreena tutorials. Don't forget to like and share my videos so that I can reach to more and more learners. Let's get started. So my dear students, what are these polysaccharides? As far as our previous knowledge is concerned, which I've shared with you all, I've told you that a polysaccharide is something in which you have a chain of monosaccharides. So it'll be something like this. If this circle is representing a monosaccharide, so in case of polysaccharides, you'll be having a number of these monomers, a number of these sugars connected to one another, and the number is going to be more than 10, and it can reach to up to thousands in range. Okay, so such long chain of monosaccharides is your polysaccharide. It's very simple. Now polysaccharides are also called glycans. Okay, so you have another term for them. They are also called glycans, and these polysaccharides have a general formula. The general formula for representing polysaccharides is CnH2O n minus 1 times. So this is the way in which you can represent a polysaccharide. Okay. Now you have this information till now, which is clear to you people, that this bond which connects the two sugars, this bond is which bond? Glycosidic bond. Okay, these things we have cleared till now because you are aware about disaccharides and oligosaccharides. In those cases, we have discussed this bond formation, so we are not going to go back to the bond formation. We know these basic things. Now, as far as polysaccharides are concerned, my dear students, you'll be broadly having two types of polysaccharides. Number one type of polysaccharide is going to be a linear polysaccharide. I'll use this short form for polysaccharides, linear polysaccharides. PS stands for polysaccharide. And another form is going to be branched polysaccharides. Okay, so what is the difference between linear polysaccharide and branched polysaccharide? It's very simple. If you have a long chain which is like this, okay, a long chain of thousands of monosaccharides, which is like this, a long chain, but it doesn't show any sort of branching. It's not having any branch. Then you will call it a linear polysaccharide. Simple. Okay. Examples I'll give you ahead. But if to the same polysaccharide, you will find that at different places, suppose over here, it's branching out, and it's branching out at which point another chain is getting attached to it. Okay. Say for example, at this place, it's again branching out and another chain of sugars is getting attached to it. Now what has become the case? What has happened basically? Your polysaccharide is now showing branching. Okay, this branching helps us to get an idea that we can call now this polysaccharide, we can call this bigger polysaccharide what? We can call it a branched polysaccharide. Ahead you will come across both linear as well as branched polysaccharides. Now analyze this. You know that if your chain of sugars is having two ends, okay, and suppose over here there is glucose and suppose over here there is another glucose. Say for example, I'm talking about this case. Now over here, this carbon and this carbon are participating in which formation? They are participating in glycosidic bond formation. That is, there's an indication that you don't have carbon number 1 available for any sort of reaction in this case of glucose. But in this case of glucose, this carbon number 1 is available for what? For the purpose of oxidation. Accordingly, we say that this end becomes what? This end becomes reducing in nature and this is non-reducing in nature. So same is the case with polysaccharides also. Here you will have this monosaccharide you can observe that its carbon number 1 is not available for reaction. That means this carbon number 1 is not available for oxidation. This end, that's why it becomes what? It becomes non-reducing end of polysaccharide. Whereas if you look at this end over here, carbon number 1, which is available for oxidation. That's why this end of polysaccharide represents a reducing end. So polysaccharides are also polar. They show polarity. Polarity in terms of what? The one end is non-reducing end and the another end is a reducing end. So they are also showing polarity. Now let us take up the different types of polysaccharides. Now, as far as the types of polysaccharides are concerned, I'll show you first of all two types of polysaccharides based on the composition. Okay. So based on composition, we have two types of polysaccharides. Number one is homopolysaccharides. Homopolysaccharides. So PS is the term I'm using for polysaccharides. Okay. It's an abbreviation I'm using. Homopolysaccharides, and another is your heteropolysaccharides. Okay. Now, you know polysaccharides are also called what? Glycans, isn't it? So can we call these homoglycans? Yes, we can call. So these are also called homoglycans and these are also called heteroglycans. Okay, that's informative. So the alternative term for heteropolysaccharides is heteroglycans, because on references you will find a couple of different names. Now homopolysaccharides, what are they? As the name indicates, they are going to be such polysaccharides which are made up of only one type of monosaccharide. So they are made up of only one type of monosaccharide. So they are going to be very simple. Their composition is going to be uniform. Whereas heteropolysaccharides are going to be made up of more than one type of monosaccharide. So this is the difference in the composition. These are made up of only one type of monosaccharide and these are made up of more than one type of monosaccharide. This doesn't mean that these have got 30 or 40 different types of monosaccharides. Actually, it could be something like this. They could be made up of two sugars which are dissimilar, forming what? Forming a dimer. Okay. Such dimers can be existing in the heteropolysaccharide in a repetitive manner. Like you have A sugar, B sugar, then again you will have this A sugar, B sugar, and the same process continues. Since you have these repetitive dimers in it, accordingly they do not remain homo, they become hetero. Okay. In certain cases, the case could be like the monosaccharide present in them won't be the simple monosaccharide. It will be the derivative of some monosaccharide. So it will be some derived monosaccharide. Or modification which we'll see is like it could be having a sulfate group attached to it. I mean to say this sugar could be sulfated or it could be maybe aminated. So these modifications are there. Okay, so you can see either a sulfate group attached to these sugars, you can see amine group attached to these sugars. That's why they become a bit complex and these polysaccharides thus fall under heteropolysaccharides. Now, as far as aminated sugars is concerned, you get another term for the same. Okay, you also call it what? You also call them glycosaminoglycans, because I told you right now that they have what? The sugars in these polysaccharides have got what? Amine group in them. Accordingly, you can call them what? You can call them glycosaminoglycans. So you got another term. So they are also called what? Glycosaminoglycans. Okay, so this is another term for these. Or somewhere you will find this term used for them, which is called mucopolysaccharides. Okay, so these are the gelatinous substances, slimy substances, which are actually, as far as the composition is concerned, they are actually the heteropolysaccharides. I'll give you details ahead. Let's first of all get some examples over here. For this, you can think of simple examples like starch is there. Starch, which is made up of wholly and solely your glucose. Okay, glycogen is there, which is also made up of only and only alpha D glucose. They differ somewhere. I'll tell you later on how do they differ. Other example is cellulose, again made up of only glucose. Okay, another example is your INULIN. Okay, I'm quoting this example. It's not insulin, it's INULIN. Okay, you have another example called Dextran. These are some of the examples of your homopolysaccharides. They are all made up of only one type of sugar. As far as heteropolysaccharides are concerned, you have a couple of different examples. Like the easiest one to remember is heparin, which is actually a blood coagulant. It's a heteropolysaccharide. Another easy example is peptidoglycan, which you have seen, which you have come across in bacterial cell wall maybe. And another example I can give you is this agar. Okay. Another example I can give you is this hyaluronic acid. Hyaluronic acid. Okay. Another example I can give you is keratin sulfate. Okay. Another example is chondroitin sulfate. Chondroitin sulfate. So these are some of the examples I've given you for what? I've given you these examples for heteropolysaccharides. We have to go into the detailed structure of these heteropolysaccharides also. I'll take up that ahead. Okay. Now over here, there is some other information you can grab. That is also very, very useful for you people. That if your homopolysaccharide is made up of only glucose, then it's called a glucan. Okay. If it's made up of only fructose, I mean to say if your polysaccharide, homopolysaccharide rather, it's made up of only fructoses joined to each other, then you will call it a fructan. Fructan. If it's made up of only galactose, then you will call it a galactan. If it's made up of only galactose, then you will call it a galactan. Okay, so these are the ways in which you can get this information about homopolysaccharides. Homopolysaccharides could be glucans, they could be fructans, they could be galactans. Similarly, if you have a homopolysaccharide made up of xylose, you will call it a xylan. You usually come across these terms in references where you get confused that what is a xylan, what is the difference between xylan and xylose. They're very simple. If the monomer is what? If the monomer of a homopolysaccharide, monomer I mean to say the individual unit. If the monomer is this, the name of the homopolysaccharide is, the name for homopolysaccharide is this. Very simple. If the monomer is glucose, the name for homopolysaccharide is glycan. If the monosaccharide is monomer or monosaccharide is fructose, the name is fructan. If the monomer is galactose, the name for the homopolysaccharide is galactan. If the monomer is xylose, the name for the polysaccharide, homopolysaccharide is xylan. So this is also very informative. Now, before getting into the depth of these examples, we should first of all get some idea about another system of classification of polysaccharides. Let me take you to that. So, let us take up the classification of polysaccharides based on the function. Okay. So, earlier I gave you this classification based on composition. Now, you just take the classification of polysaccharides based on their function. So, on this basis, we have three categories. Okay. First category is called storage polysaccharides. Second is called the structural polysaccharides. Third category are your well-known mucopolysaccharides, or what do you call them? Glycans. You have other terms also. For the time being, let us at least be familiar with these. Now, as far as the name indicates, storage polysaccharides are going to be what? They are going to be the reserve. They are going to be the reserve carbohydrate in either in plants, you can talk about broadly, or in animals. So whatever is the reserve food, reserve polysaccharide which remains in plants and animal cells, it's called storage polysaccharides. In plants you will find it in the form of starch, and in animal cells you will find it in the form of glycogen. This is one of the difference between starch and glycogen, that both of them are actually polysaccharides, both of them are rather homopolysaccharides, you know that, okay? But the difference is that you will find this homopolysaccharide in starch form in plants and glycogen form in animals. They differ from each other in terms of their branching patterns. I'll show you that ahead. As far as structural polysaccharides are concerned, name indicates that they are going to be the structural component. So these polysaccharides are components of the structure of an organism. Say for example, I'll give you an example for this. Example is your cellulose. Think about cellulose. I earlier told you that this is a homopolysaccharide. It's only made up of alpha D glucose. But where do you find the cellulose? You find it in which cell? You find it in plant cell. Rather, you find it in cell walls. So what is basically cell wall? Isn't it a structural component of a cell? The cell wall is composed of cellulose. The cellulose becomes the structural polysaccharide. Okay. Similarly, you have peptidoglycan. Okay. Peptidoglycan, which is made up of N-acetyl glycosamine and N-acetyl muramic acid. It's a hetero polysaccharide. It's not a homopolysaccharide. So you have this structural polysaccharide, could be homopolysaccharide or heteropolysaccharide. Okay. Peptidoglycan forms the structural component of your bacterial cell walls. So peptidoglycan is one example of your structural polysaccharide. Cellulose is another example of your structural polysaccharide. One being homo, another being heteropolysaccharide. As far as mucopolysaccharides are concerned, my dear students, I have earlier told you that these you will find in diverse organisms. Okay. Generally, if we talk about them in general terms, how we can remember these mucopolysaccharides, you can remember okra, what you call bhindi. Okay. Bhindi you must have observed at home. When you cut it, slice it, you get some slimy substance out of it. Okay. That slimy, let me write down over here, that slimy gelatinous. That slimy gelatinous material is basically the mucopolysaccharide. Okay. What is the function? You will find only in okra. There are multiple examples. Like you will find these mucopolysaccharides even in your own connective tissues. Okay. So animal connective tissues also contain mucopolysaccharides. Okay. Your plants, various types of plants, even for that matter various algae, various organisms which have slimy layer outside their body, they are basically, those slimy layers are basically the layers of mucopolysaccharides. Even our connective tissue contains these mucopolysaccharides. What is the prime function of these mucopolysaccharides? Keeping these two things in consideration is that they help in water retention. Okay, their job is to help in water retention. They help in retaining water, and simultaneously what they do is they help in lubrication also. Okay, this is, we are speaking this in general terms. It doesn't mean that they only perform water retention and they do this job of lubrication. They have diverse, functions to perform. But generally, if you find them somewhere, their role is basically these two. Examples for this category I have already given you. Let I quickly remind you of those. I have given you example of chondroitin sulfate. I have given you example of keratin sulfate. I have given you example of heparin. I have given you example of hyaluronic acid. So I have given full forms ahead before this. So these are some of the examples of your mucopolysaccharides. Even you can add up pectin to this collection, and even you can add up hemicellulose to this collection, which you find associated with the cell wall of plants. So these are some of the examples of mucopolysaccharides. Now let us take up these examples in detail. Let us first of all begin with the starch. Let us analyze the structure and other technicalities associated with starch. Let us take up this starch in detail. Okay, so we'll begin with starch as a polysaccharide. So, as far as starch is concerned, my dear students, remember first of all that it's also called amylum. Okay, it's also called amylum, and you will find this starch exclusively in plant cells. Okay, in plant cells you will find this starch as a reserve carbohydrate. It's a reserve carbohydrate form which you will find in plants. And in which part of the plant you will find it? Specifically talking about, you will find this starch in chloroplasts, which is the organelle. At cellular level you will find it in chloroplast, and you will find it in plastids called amyloplast. These are the two places where you will find these starch. And starch is present and in the form of grains. Okay, we call them starch grains. These starch grains can be present in your seeds of a plant. They will be present in fruits of a plant. You will find them in seeds, fruits, or you will find them in tubers, which are the modified kind of stem of the plant. So these are some of the examples where you will find starch present as a reservoir form of carbohydrate. Now, trying to understand more about starch, what is it made up of, my dear students? So, what is the composition of starch? What is it made up of? It's made up of these monomers. It's made up of alpha D glycoses. Okay. So, what is starch? It's a polymer of alpha D glucose. Now, this starch has two forms of glucose polymers. Let me make you understand what I mean to say is that now your starch has what? It has alpha D glucose molecules connected one to another, something like this polymer. So this is now what? Each dot represents alpha D glucose. Okay. So these alpha D glucoses are connected to each other. I will tell you later on. It's basically alpha 1,4 glycosidic linkage, which we know. Now this thing is called what? It's called a polymer. Okay. In starch, you have two types of such polymers. So these polymers you will find in starch in how many forms? You'll find these polymers in two forms. Let me tell you what these two forms are. So one form is called amylose, another form is called amylopectin. Okay, you have two forms. Two forms of what? They are not the forms of starch. They are the forms of the polymers which make your starch. Okay. So you have two forms of the polymer. One polymer is called amylose. So there are differences between the two. Another polymer is called amylopectin. Very important. Okay. Amylose, my dear students, it forms about 20 to 30% of the starch. Okay, and amylopectin forms about 80 to 70% of your starch. Now, one thing is to be important, you must not be confused. Both of them together form your starch. So, starch is having two types of polymers of glucose. One type of polymer is amylose, another type of polymer is amylopectin. I will give you details ahead. Now, let us take up amylose. Let us get some information. Amylose is something which is comparatively uniform like this. It's a linear polymer. Okay, it's a straight polymer, linear polymer. You will not find any branching. So it's totally what? It's totally unbranched. Something like this. But you will not find it in this orientation. It's present in starch in a helical form. So we'll write down that it is helically twisted. Okay, the orientation of amylose in starch is something like this. You can now guess what is it made up of basically. It's made up of what? Alpha D glucoses. I hope clear to you people. It's very simple. Okay, now the question is, ma'am, how many alpha D glucoses could be there in this amylose? You'll be having, generally you have 1000 glucose residues or glucose monomers present in a amylose chain. Okay, so this chain generally has 1000 glucose molecules. So I have told you earlier that one end will be the reducing end, another end will be the non-reducing end. So this is going to be the non-reducing end because the functional group is not available, and this is going to be the reducing end of this starch. Okay, once you will get the detailed structure, there you will be able to predict why it is so. As far as amylopectin is concerned, my dear students, let us have a look on amylopectin. Amylopectin is opposite to this. Amylopectin is not going to be, only linear. Although it will be like this, but it will show now branching. So if you have a polymer like this, a polymer of alpha D glucoses, now over here it is branching, you have another polymer. Okay, over here it is branching, you have another polymer. This is again branching, you have another polymer. So what is it? It is branched polymer. What is it? Branched. Very good. And if you look at it, it is having about 2000 to 2 lakh glucose monomers. Okay, so the number of the glucose monomers is more in what? It's more in amylopectin. So what is the peculiarity? They're same, but the difference is this one is linear, this one is branched, this one is helically placed, but this is not helically placed. You do not have this now. This polymer is not helically arranged. It will be somewhat similar to the same structure. Like you will have this polymer like this. Okay, it is not going to be linear. Now let us analyze these structures further a bit. Okay, let me take you about the definite structure of amylose and amylopectin. We will do that over here, keeping this information in consideration. Okay, think about alpha D glucose molecule. I will not be showing you the entire structure of alpha D molecules because that will be hectic. You have already this information that what are the things associated with alpha D glucose. Over here I will show something like this. So my dear students, if this is a simplified version of showing alpha D glucose is like this. Okay, so this is your carbon number 1, this is your carbon number 2, 3, 4, 5, and over here you have CH2OH, which is your carbon number 6. This is the basic thing. Now let us try to figure out what amylose has. Amylose is a polymer of what? It's a polymer of alpha D glucose. Now the thing is, the question is, which type of bond? Nothing to worry about. It's something like this. Let us take another glucose. Let me quickly show you the bond also. So over here you have like this, and this is your glycosidic bond. Okay, let me show you another. It's like this. Then you have another. So in this way the chain continues. You can guess about this bond, which is over here. This is which one? This is alpha 1 to 4 glycosidic bond, isn't it? Try and analyze it for at least some time, maybe for some seconds. Just try and analyze this. This is your carbon number 1 of this glucose. This is this carbon number 4 of this glucose. Carbon number 1, let me number it, 2, 3, 4, 5. I have trained you for this purpose in my initial lectures. This is carbon number 1, 2, 3, 4, 5. Okay, if we complete this, over here you have CH2OH, okay, and over here your H. I don't need rest of the things, okay. Where is your glycosidic bond? This portion is your alpha 1,4 glycosidic bond. Same as the case over here. This is again alpha 1 to 4 glycosidic bond. How is it formed? That you people know. Now let me try to finish it. Why I am completing? Because I have something to do with it. CH2OH and H. So similarly the chain continues. You have one glucose joined to another glucose joined to another glucose in a linear manner. Till how many? Roughly about how many glucoses? You have roughly about 1000 glucoses connect to each other with the help of these alpha 1,4 glycosidic bonds. That chain is going to make your what? That is going to represent your amylose. So which type of bond in this case between glucoses? You have alpha. Very good. You have alpha 1 to 4 glycosidic bond. Very simple. This over here is the representation of what? It's the representation of amylose. Very simple. Simplified manner is to show like a chain. Glucose, 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 glucose. And you can show this chain like this. Okay. So this is the simple representation. If you want to look at the structural part, I have shown you. Now this thing is not present in starch like this. You will find it in the form of a helix. So this is the real form of your amylose. But technically at structural level it is something like this. Now my dear students, think about amylopectin. Amylopectin also has the same polymer. But the difference is very simple. Let me take another molecule of glucose over here. I will draw another molecule of glucose. Okay. Suppose this is another molecule of glucose. Over here this is oxygen. Clear? Okay. Let me complete this. This is CH2OH. I hope it's clear. It must be clear because you are familiar with these Haworth projections. I have told you everything about alpha D glucose before in my previous lectures. So this is carbon number 1, 2, 3, 4, 5 and 6. Okay, so again the same thing. Let me show these lines also. Now the thing is, like, you will have the same chain. So you will have to this glucose, another attached glucose, another attached glucose. So what I am trying to tell you over here is that you have a long chain of glucoses in this case also. So to make it very simple, I will show like this. Suppose you have over here another kind of thing like this. Okay, so this is like this. What I mean to say basically, okay. So over here this is also joined to another glucose, that's also joined to another glucose. So you have one amylose chain, you have another amylose chain. Over here this is also 1,4 linkage, okay. This is carbon number 1, carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and you have carbon number 6. So this chain I'll show you with the help of dots. It's a continuous chain, okay. This is also a continuous chain. So this represents one amylose chain. Over here you have another amylose chain. You have 1,4 bond here, 1,4 bond here, 1,4 bond here. So you have how many amylose chains right now on the board? You have 2 amylose chains on the board right now. One on the top, one on the bottom. Okay. Now see what they do. They prefer, in case of amylopectin, they prefer creating a bond between the two. Now, which is forming the bond? This carbon number 6, which is present over here. Look at this. I will place my pen over here. This is carbon number 6. And this over here is carbon number 1. You have what? You have H and OH. I hope you remember. Okay. Now see what they do. They make a contribution in which this glucose releases this OH. And this glucose, which is there in this chain, I'm talking about this glucose, it releases its H. What is being done? Dehydration. Which bond will be formed? Glycosidic bond. So something like this will happen now. Let us remove this OH. Okay, it's gone. Let us remove this H. It's also gone. Okay, so H and OH gone. Water molecule is gone. Now what will be the product? O is left. Okay, this O, I'll remove this O and I'll place this O over here. Look at the board. I'll place this O over here. See what is the difference I made? What do you got over here? What do you get over here? This is a glycosidic bond again. A bond between COC. Over here is a bond between COC, glycosidic. This is also COC, dot represents C. So this is also a glycosidic bond, isn't it? Glycosidic bond, very good. But this bond is created between carbon number 1 and carbon number 6. Analyze. You have carbon number 1 participating and carbon number 6 participating, my dear students. So we'll call it alpha, because it's alpha sugar. 1 to 6 glycosidic bond. Okay, that is the difference between amylose and amylopectin. I will try to encircle your amylopectin completely with blue color. So I will be encircling this. Okay. This thing which is on the board right now, which I am breaking out with the help of blue color pen. This structure is, my dear students, your amylopectin. Okay, this is your amylopectin. So what do you observe? You observe that amylose is made up of, it's a linear polymer. So how many amyloses you have? 1 and 2. This is linear and you have only alpha 1,4 bonds in it. Amylopectin on the other hand has branched. One polymer, another polymer, they are branched. So this is branched, having both alpha 1 to 4 linkage as well as alpha 1 to 6 linkage. Alpha 1 to 6 linkage is present where? It's present near, the branching point. Exactly. So this is entirely now pure starch. So starch is made up of what? Let's analyze. Starch is made up of what? It's made up of alpha D glucose. Okay. Do you find alpha D glucose? Everywhere it is alpha D glucose. Clear? Starch is a polymer of alpha D glucose. Yes. See, 1, 2, 3. chain is basically polymer. I have shown dots, 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 so you have many such sugars. So a chain of alpha D glucose is a polymer, okay? But how many types of polymers you see in starch? 2 forms of polymers. One form is amylose, which is linear, branched or unbranched. It's unbranched. Amylose is unbranched, okay? And having only which bond? Alpha 1,4 linkages. Amylopectin, on the other hand, is branched, which I have shown you completely. It is branched, so you have one polymer branching into another polymer. You have both alpha 1,4 linkages as well as alpha 1,6 linkage. So this linkage is which linkage? This linkage is which bond? Alpha 1,6 bond. So this is branched, this is unbranched. This linkage is present only at which point? This is present only at branch point. So this is going to help you to understand the starch both for chemistry as well as for biology. Now let us take up, my dear students, let us go to the glycogen. Let us try out glycogen over here, okay? Let me go to glycogen. Now, my dear students, if you observe, I have not erased this amylopectin. There is a reason I have not erased. I will tell you why I haven't erased. Let us take up the second type of homopolysaccharide, that is glycogen. Let us understand more about glycogen. Now, the first thing which you need to know about glycogen is that you will find this as a reserve carbohydrate in animal cells. This is the first important part. So this you will find in which cell? Animal cell. This is the major difference between starch and glycogen. So where do you find in animal cells? Where will you find glycogen abundantly? You will find this in especially liver cells. Okay? Because it's in the liver cells that glucose is basically stored in the form of glycogen. So you will find more glycogen droplets in which cells? In liver cells. Okay? It's not only liver cell. In fact, your muscle cells also contain a large amount of glycogen. Okay? Because energy is stored specifically in these two types of cells, liver cells and muscle cells. Even you will find the same glycogen as a reserve form of carbohydrate in case of bacteria and fungi. Okay, so bacteria and fungi also store glucose in the form of glycogen. You might be knowing about these terms that when you digest carbohydrates in your digestive system, when glucose is sent to your blood finally after absorption. So suppose this is the glucose which is present in actually your lumen of small intestines. Finally, this glucose is being absorbed and transported into what? It's transported to the bloodstream. And from bloodstream, it's taken up by your body cells. This is the basic thing which you know about absorption. You know, in case of body cells, if you talk about liver cells, what is happening near liver cells? In case of liver cells, glucose is again polymerized into glycogen. Okay? So glucose is stored in your body in the form of glycogen. This process is called glycogenesis. It's called glycogenesis. Okay? And once you need glucose, that means during starvation and other conditions, during the month of Ramadan when you are fasting, at that point of time, glycogen again, it breaks down, it starts breaking down in your cells, releasing what? Releasing glucose back. Okay? This process is called glycogenolysis. Okay? So you have two processes over here. One is glycogenesis, which is the formation of glycogen in liver cells, and another is glycogenolysis, which is the breakdown of glycogen to release glucose back. This is generally happening in your body as far as the metabolism of glycogen is concerned. Okay, this is also very important to remember. Now the question over here is that your cells are storing what? Your cells are storing basically glucose in the form of glycogen. When we talk about which cell? When we talk about animal cells. In plants, the same glucose is stored in the form of starch. Okay. Now, going ahead, how does this glycogen appear? Like, how is it structurally composed of? Is it linear or branched? You will find that glycogen is basically, it's a highly branched polymer of same alpha D glucose. So as you know, your starch was what? It was made up of two things, amylose, plain chains, and amylopectin, which were branched. Glycogen is not made up of two types of polymers. It's again made up of alpha D glucose, but this alpha D glucose is absolutely present in which form? It's present in branched form. That means a form which is similar to amylopectin. Okay, but what is the difference? Is that it's highly branched. It's not as branched as in case of amylopectin. It's highly branched. Like this, in its form, you will see some protein in the center first of all. To this protein, what are attached are the polymers of glucose. So I'll show, like, this is one polymer of glucose. This is a chain which is made up of maybe 100 or 200 glucose molecules. Then it branches out like this. Again, this also branches like this. Again, you have another chain which is also branching. Then this is also branching. Then you have a chain which is again branching and branching. Finally, you have a chain which is again branching. Can you observe what is happening in this structure is that the structure is becoming what? It's becoming more and more globular, round shaped, and it's highly what? It's highly branched. Okay. This type of a structure is shown by your glycogen, whereby what is happening is basically you are getting short chains, they are getting branched, this branch is again branching, that branch is again branching, and finally you get a structure similar to this in case of glycogen. So you do not have those helical chains, you have this globular structure of your glycogen. And if you want to remember that in case of amylopectin, you have branching taking place after every 24 to 30 glucose molecules. Okay. So if you have 24 to 30 glucose molecules attached, then a branch arises. Okay. But in case of glycogen, what is the case is that after every 8 to 12 glucose molecules, after every 8 to 12 glucose residues, you will see what is arising is a branch. So branching is frequent in glycogen. Suppose over here I had made a branch, okay, I had made a polymer. It's how much long? It's 18 units long and it shows branching. Again 18 units long, again it's showing branching. So after every 18 to 12, 8 to 12 molecules of glucose, after every 8 to 12 molecules, means at short intervals you will find a branch. At another short interval you will find a branch. So which becomes more branched? Is it amylopectin or glycogen? Glycogen is highly branched. Otherwise the bonds are the same. In glycogen you will find the same bonds. Alpha 1,4 bonds which form the polymer and alpha 1,6 bond which form the branch. The question is you will find more and more branching in case of glycogen. So what about bonds? You will find both alpha 1 to 4 bonds as well as alpha 1 to 6 bond. The difference is in glycogen you have branch after every 8 to 12 molecules of glucose, and in case of what? In case of amylopectin, branch arises after every 24 to 30 glucose molecules. Although the structure is same. So if I want to draw glycogen now, it will be same. I will be showing glucose, 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 a long chain attached with the help of alpha 1,4 linkage. Again branch. You have this, then maybe this will again branch, and that will again branch, and another branch will again branch. So that is the case in which the two differ. There is one more thing which you can remember over here is about the reaction. The reaction of glycogen with iodine. So if you take glycogen and you allow it to react with iodine solution, the color which will be visible, the color which you will get will be reddish violet. Okay. So these are some detection tests. So it will be reddish violet color you will get on using which solution? Iodine solution. If you do the same test with starch same test with starch, that is going to give you black color. Okay, that's going to give you black color. This is how you can do the detection part of these homopolysaccharides, both starch and glycogen. Now there are some other examples which we will like to ponder upon before going to cellulose. I will give you slight information about some other homopolysaccharides. Till now we are done with the storage polysaccharides. Let me take you again quickly. So I have given you storage polysaccharides. Out of storage polysaccharides, my dear students, we have thoroughly gone through number 1, starch. Okay. Number 2, we have discussed everything about glycogen. Okay. Now, as far as your competitive examinations are concerned, you could be asked questions pertaining to some other storage polysaccharides, like you could be asked about INULIN. INULIN it is. Okay. INULIN, my dear students, is a fructan. It's a fructan. I hope you remember this term. I've given you this term initially. Fructan is a homopolysaccharide which is made up of fructose exclusively. Okay. Inulin is made up of more than 20 fructose monomers or fructose residues. So you have more than 20 fructose molecules present in this inulin. Now inulin, these fructose molecules need to be bound to each other. They are bound again with the help of glycosidic bonds. Now, which type of glycosidic bond you will see over here? In case of this fructan, you will see beta 2-to-1 glycosidic bond in this case. Okay. Now, why is it so important? Because inulin, my dear students, is used during a procedure which is called dialysis. Dialysis is a simple procedure. Inshallah, we will discuss it once we will take excretion in humans. Dialysis is a procedure which is given to a person or a patient whose function is not functional. So in this procedure, what is being done? Blood is filtered and blood is actually purified to get rid of nitrogenous wastes. So it's an artificial procedure. It's a procedure which is carried out by taking blood out of the body. Now we don't need to go into the details of dialysis. What is the point over here is that this compound, this polysaccharide, is used during dialysis. For what purpose it's used? It's used to measure the rate of glomerular filtration. Okay, you'll be able to understand more about it once we'll take this excretion. Glomerular filtration is actually the first step of urine formation. So once dialysis is being performed, we need to check out the glomerular filtration rate of the patient, and for checking that, this compound is used, this polysaccharide is used. So it's very important for you to keep this thing in mind, that what is the application of this inulin. The fourth thing I want you people to remember is very, very important as far as exams are concerned. The fourth item I will be giving you is laminarin. Laminarin is again a storage polysaccharide which you will find exclusively in your brown alga. Okay. So brown alga have carbohydrate or polysaccharide stored in the form of laminarin. Number 5 I will be giving you is leucosin. Leucosin is the storage form of a polysaccharide which is exclusively present in diatoms. Okay. Next I will give you is a paramylum starch. Paramylum starch. You remember starch is also called amylum. So you have paramylum starch which is exclusively present in your euglenoids. Okay. The last one which I'll be giving you is Floridian starch. Floridian starch is a type of storage polysaccharide which is exclusively present in red alga. Okay, so you have these additional examples of storage polysaccharides. We don't need to go into the depths of these. We have to remember the details about these, but I have given you additional examples also. As far as the second category is concerned, they are what? They are your structural polysaccharides. Now in this category we will be discussing, inshallah, that's our next topic. We'll be discussing number 1 cellulose in this category. Next we will be discussing is peptidoglycan. Okay. And next, after that, I'll be giving you information about this one also, which is chitin. Okay? Once we'll be done with these, then we will finally go to the mucopolysaccharides. Okay? Done? So this we will go later on, and now our next step is to touch these. Specifically, we are going to touch cellulose next. Now, over here, let us think about these terms. Homopolysaccharide and heteropolysaccharide. Which among these is homopolysaccharide and which is heteropolysaccharide? Okay. So, as far as these storage polysaccharides are concerned, all of these examples, they are all homopolysaccharides. Okay. While as if you go to structural, if you point out cellulose, this is also homopolysaccharide made up of only one type of monosaccharide. But if you go to whom? If you go to peptidoglycan and chitin, these are heteropolysaccharides. If you go to mucopolysaccharides or glycosaminoglycans, they are also heteropolysaccharides. This is very important for you people to remember. Don't ever mingle these things. Keep an eye upon what is a homopolysaccharide, rather which is the example for a homopolysaccharide and which among these is a heteropolysaccharide. Our next topic is going to be cellulose. That is a structural polysaccharide, but that is which polysaccharide? That is a homopolysaccharide. Let us go to cellulose now. So, chitin, you have to remember that the current edition of your NCERT says that chitin is a homopolysaccharide. That's absolutely correct, because chitin is made up of only one type of monosaccharide, that is your N-acetyl glycosamine. But since this monomer or monosaccharide is not a simple sugar, it's not simple, it's derivative. That's why it is also considered to be a heteropolysaccharide. But if it will be asked in examinations, it will be better to say that chitin is a homopolysaccharide. Now let us go to cellulose. Okay, now let us understand about cellulose. It's very, very interesting, very, very important also. So as far as cellulose is concerned, my dear students, number one thing you need to remember about cellulose is that this is the part of cell wall of your plants. So this cellulose is, that's why it's the most abundant organic substance present on the earth, because you have a tremendous number of plants on this earth, and every plant cell is made up of what? It's having this external covering of cell wall. That's why you say that cellulose is abundant. It's the abundant biomolecule, abundant substance which is present on the surface of the earth. As far as its composition is concerned, it is made up of another anomeric form of glucose, that is beta D glucose. So it's not made up of alpha D glucose, it's made up of beta D glucose. Another point to ponder upon. And in this case, the chain, the polymer, is not having alpha 1 4 glycosidic linkages, it will be having beta 1 to 4 glycosidic bond. Okay. Now, I hope you remember that I've told you amylose, which I've shown you in case of starch, that is in the form of a helix. Okay. The reason amylose appears in the form of helix is because it's made up of alpha D glucoses. Okay. And whenever two alpha D glucose molecules form glycosidic linkage with each other, they get bent, due to which the structure finally becomes helical. So the final amylose won't be straight or linear. It will be helically arranged. But if you look at cellulose, in case of cellulose, you have what? You have beta D glucoses. Okay. So the speciality over here is that once these beta D glucose molecules are attached to each other, the final product is fully stretched form. Okay. So it's fully stretched. That means the chains of cellulose are linear. That's very important as far as the high tensile strength of cellulose is concerned. Let me make you understand. So you need to remember that amylose is not linear finally. Okay, amylose finally it turns out to be a helical structure because of the bending of alpha D glucose molecules during glycosidic bond formation. But here beta D glucose molecules, once they form the bond with each other, the final product becomes linear or fully stretched. Let us understand it properly. Let me now make you understand about it. So what is the nature of cellulose? Is it branched or unbranched? So cellulose is completely unbranched. You will not find any branch in cellulose. It's unbranched, and you will find it to be linear. A linear polysaccharide, which is made up of around 6,000 glucose residues. So you will find 6,000 glucose molecules present in a cellulose chain. Now, trying to understand it more, let me show you the linkage which is present in cellulose. So I'll be going with one beta D glucose. So in case of beta, you have this anomeric carbon in different orientation. You will be having now OH towards the top and H towards the bottom. So over here I'll be showing you the final products directly, in which I'll be placing these glucose molecules attached to each other with the help of the linkage which is there in the cellulose. So in case of cellulose, you will find that this is carbon number 1, carbon number 1, carbon number 1, this is carbon number 4, carbon number 4, carbon number 4. So this form over here, you have to think that it's the beta glycosidic form. So the bond formation will be like this. You will be having something like this over here. So you have an oxygen bound to this. Okay. Over here, the same case, oxygen bound to this. So this is the case. Here you have H and here you have H. Okay. Here you have H and here you have H. In this way, these beta D glucose molecules are connected to one another. The bond, the linkage between two glucose molecules is beta 1 to 4 glycosidic bond. Beta, because this glucose is not in alpha conformation, it is a beta anomer. So that's why this is what? This is beta D glucose. That's why the linkage is beta 1,4 glycosidic linkage. And these linkages are responsible for keeping the structure straight. Okay. So a cellulose chain is like this. It's a straight chain. Okay. Now, as far as the cellulose, which is there in the cell wall, there is a proper arrangement of these cellulose chains. Now, you will see, suppose this is one cellulose chain. This is another cellulose chain. This is another cellulose chain. This is another cellulose chain. Observe these cellulose chains at different locations. Suppose over here you have CH2OH. Okay. Suppose over here you have H and OH. So that means at different locations, what do you have available? You have these OH groups available. What are these OH groups called? They are called hydroxyl groups. And from chemistry, you know, wherever you have these hydroxyl groups in close proximity of each other, that can result in the formation of hydrogen bonds. The same happens over here also. So suppose this is one cellulose chain, this is another cellulose chain, this is another. So I've shown 4 cellulose chains for the time being. Each cellulose chain is made up of thousands of beta D glucose molecules attached to each other with the help of beta 1,4 glycosidic linkage. That's clear. Now, once these cellulose chains come closer to each other, they interact with each other by means of their hydroxyl groups. So consequently, what is being formed between these cellulose chains? You will get these hydrogen bonds formed between the cellulose chains. Observe this carefully. So these hydrogen bonds are very, very important. As such, cellulose chain is not so strong. It's a very fragile chain. But once these cellulose chains interact with each other, they become held with each other with the help of these hydrogen bonds. They become tough. Okay, this structure will definitely become tough. What is this blue color dots? They represent your hydrogen bonds. And these bonds are made because of hydroxyl groups present in adjacent chains. They are interacting with each other and you have this hydrogen bond formation. Now this arrangement over here, this arrangement in which you have different cellulose chains interacting with each other, this arrangement makes a group which is called, suppose this fiber is called a microfibril. Okay. So when you have many cellulose chains attached to each other with the help of what? With the help of hydrogen bonds, you call this arrangement a microfibril. What do you call this arrangement? You call it a microfibril. So you have thousands of such chains present in a microfibril. Now what will happen is, these microfibrils, many of such microfibrils will join with one another. They will form a bigger fibril. Say for example, I'll show over here a big fibril which is comprised of what? Which is comprised of many such microfibrils. Many number here indicates hundreds, okay? You can have around about 250 such microfibrils now. So you have around about how many? 250 microfibrils getting arranged together, forming a bigger fibril. This bigger fibril is your macrofibril. Okay? This is your macrofibril. So I have less space over here, I'll write down here. So this is a macrofibril. Okay? Now when you talk about the actual cell wall of plants, in them you will find, so these microfibrils are arranged like this. Okay? They are arranged in different directions. See? You have some microfibrils arranged vertically, some microfibrils arranged horizontally, some microfibrils arranged diagonally. So in every geometrical position, these microfibrils are placed. So each microfibril is itself made up of what? Each macrofibril is itself made up of microfibrils. Each microfibril is itself made up of cellulose chains. So what are these representing? These are representing your cellulose chains. Okay, so you can see now that this is the condition with what? This is the condition with your cell wall. You can see that how much strength will this cell wall be having? It will be having high tensile strength. Be having high tensile strength because it's made up of so much of the tough material which has got its toughness from what? It has got its toughness from these hydrogen bonds between the individual cellulose chains. Further, these chains are getting compact in the form of microfibrils. They are further compacting in the form of macrofibrils, and those microfibrils are getting arranged in different sorts of orientations, giving rise to the actual cell wall of a plant. So that's why we say that cell walls are very rigid and very tough structures. Okay, where do you find these cell walls basically? Say for example, if you take a plant cell, okay, suppose this is a plant cell, and this portion is the cell wall which I have made with blue color. Inside you have cell, cytoplasm and everything. I'm not concerned with that over here. And externally, what is present in plant cell is the cell wall, which is made up of cellulose. So this cell wall is having high tensile strength. It's having high tensile strength means that suppose if water will enter into this cell, okay, this cell is not going to burst as is the case with animal cells. If water enters osmotically into a animal cell, there is a possibility that finally the cell wall will burst. But that will not happen with the plant cell because of its cell wall. This plant cell will take up the water and finally there is a stage it will become turgid. Okay, it will become fully turgid where the cell wall will not allow it to burst. It will not allow the cell to burst. It will resist the pressure. It will give the opposite pressure to the expanding cell, not letting it burst. In that manner, it gives a kind of structural support. It gives a kind of strength to the plant cell to bear this pressure and to hold this overall body of the plant. So this is one more important aspect of this toughest polysaccharide, which is called cellulose, which forms the cell wall of plant cells. Now there is another question, another thing we can discuss over here, which is there in your syllabus also. That is regarding the action of enzymes. The question is cellulose digestible or not? If we talk about us, animals, or specifically about humans, we cannot digest it. We cannot digest cellulose. Why? Because the enzyme which is responsible for digesting cellulose is cellulase. Okay? This cellulase is an enzyme which breaks which bond? It breaks beta-1,4 glycosidic bonds. Because we do not have cellulase, it's not possible for us to digest cellulose. Okay, so if we are taking cellulose along with the diet, we are not getting any sort of energy from that cellulose because our body is not capable of digesting it and getting out individual glucose molecules. We lack these beta bond attacking enzymes. Specifically in this case, we lack cellulase enzyme. Okay, but this is very important. This cellulose is very important because it forms a part of our dietary fiber, which is very important for maintaining the gut health, which is very important for preventing constipation and other problems. But if you look at certain animals, I'll rub it off. If you look at animals like ruminants, if we talk about cow and sheep and all, so let me talk about ruminants here. Out of ruminants, I'm talking about your cattle, I'm talking about deer, I'm talking about giraffe maybe, okay? I'm talking about these animals. So what do they have, my dear students? These ruminants have special bacteria in their gut. Okay, they have special bacteria in their gut. They are not harmful bacteria, they are actually symbiotic bacteria which live in their gut. Now what do these bacteria do? They produce the enzyme cellulase. And because of the cellulase produced by the bacteria in their gut, they are able to digest cellulose. So that's why cow and sheep, because they are wholly solely herbivores, they can digest cellulose with the help of these symbiotic bacteria which are there in their gut, which secretes cellulase enzyme. Okay, so they are not themselves producing this enzyme, it's the bacteria which is living in their gut, they secrete this cellulase enzymes. For that matter, even termites also have symbiotic bacteria in their intestines. Okay, which help these termites to digest cellulose, because those bacteria also release cellulase. So some of the animals like ruminants or your termites, ruminants including your cattle, deer and giraffe, they are capable of digesting cellulose with the help of this enzyme cellulase, which they are not producing themselves, which is being produced in their body by the symbiotic bacteria. Those bacteria are giving them cellulose and which is aiding in the digestion of cellulose in these particular organisms. So as far as this information is concerned, this is more than sufficient for you people in this particular class. So cellulose abundantly present, and there's one more thing you can remember. Like if you think about cotton fiber is 90% cellulose. Okay, and if you look at your cell walls or the plants, they are majorly made up of cellulose. Now in cell wall, it's not only cellulose, you will find some other substances which we will discuss under mucopolysaccharides. For the time being, cellulose is a homopolysaccharide. That too, it falls under structural polysaccharide. And one of the most important thing is that you will find the cellulose in a straight form. Many such cellulose chains are joining each other with the help of hydrogen bonds, and they are forming a structure called microfibril. Many such microfibrils form macrofibril. Macrofibrils are arranged in different sorts of orientations which I've shown you earlier, that result in the highly rigid and supportive and high tensile strength structure which is called your cell wall. So this was all about cellulose. Till now we have completed our portion till cellulose. So since the duration of this video is now exceeding 1 hour, so the rest part, the rest component of your polysaccharides, specifically your homopolysaccharides, they are done in this particular portion. As far as heteropolysaccharides are concerned, inshallah, I will break this lecture into 2 parts and part 1 will be having information till this point, and in part 2 I'll be sharing this information about peptidoglycan and chitin and all the mucopolysaccharides. So you will get details about rest of the components, rest of the parts in part 2. This was all for this video lecture. Let you wait for the next video and let you exploit this video, let you exploit this information which I shared with you all in this particular video lecture. Don't forget to like, share and subscribe my channel. Don't forget to share it as much as possible so that I can reach to more and more learners. Till then, take care of yourself. Fi Amanillah.